analyzing some single-cell RNA-seq data in R, and I will be doing it in Jupyter Notebook, okay? So first, I will be giving some theoretical background. I will be explaining the differences between RStudio, Jupyter Notebook, and Jupyter Lab for the ones who don't really know the difference. And then I will be giving like or explaining the different steps in a single-cell RNA-seq analysis pipeline. And in the end, we will be analyzing some study case, which is this one here, which is from pancreas cancer. And we will be doing some clustering, markers identification, cell type annotation, and differential expression analysis. So first, just to clarify these terms, R is the programming language, and normally people work with R in RStudio because the interface of RStudio is more user-friendly. So if you're a beginner, you normally use RStudio. But if you are a bit more advanced, maybe you tend to use either Jupyter Notebook or Jupyter Lab because they provide more advanced specifications, but it's just a different interface to run R, okay? It doesn't matter where you run it. But today my preference is working with Jupyter Lab, okay? So I will let this slide here for the people that are interested on trying Jupyter Lab for the first time, okay? So you should follow these steps. But let's get started with the pipeline, okay? Or the workflow of single-cell RNA-seq analysis. So normally we start with a sample. It can be any type of sample you are interested on studying, maybe tumor sample, even or a plant tissue. So we normally extract the RNA of this sample, and the next step is the crucial one in single-cell RNA sequencing analysis, and is marking every transcript. So we use two kind of markers or barcodes. One is a cell barcode, which is specific to the cell where the transcript belongs to, and the second barcode is a transcript barcode and is specific to the transcript. So this is the crucial step that allow us to know from which cell is every transcript coming from. So then we amplify all these transcripts and we get a count matrix. So this is the basic principle of single-cell RNA-seq sequencing and is what allows us to know the gene expression of each cell, as opposite to bulk RNA sequencing. In bulk RNA sequencing, because we don't mark, we don't have these cell barcodes, we don't really know from which cell the expression is coming from. We have an average gene expression. So maybe let's get started with the practical part. We will be analyzing this study here, this pancreas cancer study. I will show you guys this study first. So I cannot really see. Wait. Yeah, okay. So this is the study. And they basically provide 32 samples, okay? They have samples from donors, which are healthy pancreas samples. They have samples from the tissue surrounding the tumor in pancreas cancer, and they have samples directly from the tumor. So they have 3 different types of samples. So from this study, I chose one sample from the donor, so a healthy sample. I chose a second sample around the tumor and a third sample directly from the tumor, okay? So basically, the first step in the pipeline is normally performing a quality control of each of the samples you want to study separately. And after performing the quality control, you perform the integration of the samples. So I have already done this quality control and integration because it's kind of a long process. So we will start today from the integrated data, and we will be doing the downstream analysis, which is the most interesting part, okay? So as I said, I've done this quality control and this integration. And the first thing we will be doing is the dimension reduction of the data. So let me show you the code. Well, by the way, guys, I want to provide you with the code, okay? So I have here my GitHub. I have here the code for the Jupyter Notebook and the code for RStudio and RScript. So maybe it's a good idea that I share it in the chat, okay? So you guys can run it later at home. Where can I? Yeah, chat. Okay, so there, guys, you can download the code and run it anytime you want, but today just focus on understanding the different steps and how the pipeline is formed. So the first, here I have the R notebook, okay? And the first step is loading all these libraries that we will be using today. The most important package is this one, Seurat, which is a package in R that allows us to analyze the single-cell RNA-seq data. So if you have not downloaded these packages yet, you have to download it first and then load them. So I will load it now. And then the next step is reading the integrated data, okay, that I will provide it to you later. So this is the integrated data with the 3 samples. And after reading the data, we have to set the default assay as integrated, meaning that we will be using this integrated data already normalized. So the next step is scaling this data. We need to scale the data because there are some genes that are higher expressed than other genes. And in order to make all genes comparable for the analysis, we need to set them to the same scale, okay? So we have to scale the data. And the next step is the dimension reduction step. So So in single cell, we have thousands of cells and thousands of genes. So we have a really high dimensional data set. So we need to reduce the dimensionality of this data. And for that, we will run a PCA, a principal component analysis. So with this principal component analysis, we will get different principal components, which are in this case linear combination of genes. And the first principal component that you will get is the most informative one, is the one explaining most of the variance in the data. So that is the first thing we will do now. We will run this PCA, okay, in our integrated data. And we will be getting the different principal components. It takes some seconds, but it shouldn't take too long. But once we get these principal components, we need to choose which number of principal components is the best for our data. Here we have the results. So here it is showing the first 5. And we see that, for example, in the first principal component, there are genes with a positive effect and genes with a negative effect or contribution to the linear combination, but all of them are really important and explain most of the variability in the data. So as I said, we need to choose the right number of principal components for our analysis. And this elbow plot here that I just ran is showing in the x-axis the number of principal components and in the y-axis the standard deviation they explain. So basically, we see it has like an elbow shape, and we see that from the first 40 principal components, the rest of them, they don't really explain any more variability in the data. They are not informative anymore, just the first 40. So this is the number we need to choose, 40 first principal components. This number will depend in your data, and that's why this plot is important to look at. So then, after selecting the right number, we need to run the UMAP. So what is a UMAP? UMAP is also a dimension reduction technique that the difference is that it's a non-linear method. So it actually helps us to preserve the local and global structure of the data more than the PCA. So that's why we have to run it now. And we have to set the dimension reduction method to PCA. And we have to indicate the number of principal components we have set before. So we said this 40, so we have to set this to 40. Now it's running. It takes some time as well. But after running this UMAP, the next step is to find the neighbors. So cells that have a similar gene expression. These cells will be neighbors. And this is what we will use for the clustering later, okay? So this normally takes a bit more time, but I already have the notebook already run, so we can look at it. So here I run the find neighbors, and then the next step is finding the clusters. So we run this function find clusters, and we specify the resolution. So a lower resolution will give you less clusters with a bigger size, and a higher resolution will give you more clusters with a smaller size. So the resolution you choose will really depend on the data you have and on the cell types you are expecting to see. So in the paper, in the study we are analyzing now, they annotate, I think, 13 cell types. So we need a resolution that allows us to kind of identify at least this number of clusters. So we normally start with a low resolution, and we keep increasing if we need more resolution. I don't know if this is finished. Yeah, it's finished. It's fine, we can look it here. So we run this at resolution 0.1, and then we visualize the results with a dim plot. A dim plot is a dimension plot which allow you to see the result of the clustering at this resolution. And you can see here how all the cells are clustered. And we see there are 15 different clusters. I think this is a nice resolution for this case. If you increase this resolution, you will get more clusters. Yeah.
Ma'am, slide is not clear.
This one?
Maybe, Aida, maybe you can zoom it a bit.
Okay. You mean the plot, right?
Yeah, now it is fine.
Okay. Yeah, so if we increase the resolution, for example, this cluster number 0 will be even more split. Maybe I can show how the resolution 0.4 would look like. Okay. Or maybe 0.3 for you to see a bigger resolution. But this is a bit subjective, the resolution you choose. Again, it really depends on, here's resolution 0.3. You can see now these clusters of cells is even more split. So it really depends on the number of cell types you are expecting to see in your analysis. But because we are expecting around 13 cell types, this resolution is fine. and we can even split by sample. So this would be splitted by sample. This is the healthy tissue of pancreas. This is the tissue around the tumor, and this is the tissue directly from the tumor. So here we can already see some interesting things. We don't really know yet which cell types are here. We will do it later, but we can see, for example, this cluster number 0 is less, it has less number of cells in the tissue around the tumor and in the tumor. And then, for example, this cluster number 8 is has more cells than in the healthy tissue. So we are already seeing some interesting things, but we actually have to annotate the cell types, the different cell types. So this is the same plot, but if we group it by sample, it's the same thing. Here it is splitted by sample, and here it is grouped by sample. So the next step is finding the markers of each of the clusters. So that we do it with this function, find all markers, and we give the argument only pos, which means only positive log-fold changes. So this takes more time, okay, because it has to calculate all the markers for every cluster. The more clusters you have, the longer you have to wait. But in the end, you will get a data frame with all the markers for every cluster. So for example, here in this column, it is indicated the number of the cluster. This is cluster 0. And here in this second column, you have the log-fold change of the genes. We can see the first gene is the highest, is the one with the highest log-fold change in cluster 0. So this would be the markers for this cluster. You can save this data frame this way, okay? And I have already downloaded the data frame, okay? This is the whole data frame. So this would be for cluster 0, as you can see, but if you scroll down, you will start seeing, for example, cluster 1. So this would be the markers for cluster 1, and the same for every cluster, okay? Cluster 2, cluster 3, etc. So that's how you find the clusters for each, the markers for each of the clusters. And now is the annotation part. So normally you use, you look for markers that are already known, okay? So maybe from different publications, etc. So here I have the markers that they use in this study. They use these markers, but I also have a data set, a data frame that I also have in my GitHub with many markers from many different cell types. So you need to check these markers in your data in order to annotate your clusters, okay? We will see it now. So they mainly, in the study, they mainly use this. And I will show you now, for example, these two markers, these two genes here are markers for acinar cells, okay, which are a type of cell in the pancreas. And we can see the expression of these two genes are mainly in this cluster here. That would mean these cells are acinar cells, okay? Because it is known that these genes are markers for acinar cells. Then these two genes are markers for ductal cells, another cell type in the pancreas, and we can see they are mainly expressed in this cluster here and this cluster here. Then these two markers are for endothelial cells, and we see they are expressed in this small cluster here. Then these two markers for fibroblasts, they are expressed in this one. You can clearly see how it works. So then for T cells, this is a marker for T cells. So this cluster here would be T cells. Then for B cells, these two genes are markers for it. So it would be this small cluster here. Cyclin cells, this is a really good marker for cyclin cells here, cells that are cycling. Endocrine cells, we can see endocrine cells would be just this cluster here, okay, because it's the one expressing the most. Then for macrophages, we can see all this big cluster here would be macrophages, including this one. For mast cells, it could be this cluster here, etc. So this is the way we kind of identify to which cell type the cluster belongs to. So now is the time where we have to give it the name. We have to annotate it. So here is how it is looking before annotating the data, right? As I showed you before. And now we have to annotate this cluster. So we use this function here, rename identities. So we pass the integrated data and we say we rename, for example, cluster 0 as macrophages because we saw this cluster 0 was expressing a lot of macrophages markers. Cluster 1 as acinar cells, cluster 2 as T cells, cluster 3 as, again, acinar cells, etc. You do it that way. But for example, for cluster number 14, which is this one here, we didn't see it is expressing any of the markers, okay? Sometimes that can happen, that you have a list of markers, but you find some cluster that doesn't really express any of them. So this is what is happening with this small cluster, is not expressing any of the markers. So what we can do now is opening the Excel file we created before, okay? The one, let me show you, the one for the markers of each cluster that we did before, this one here. We open it, we look for the cluster number 14, which is in the bottom. Yeah. And we look which markers, which genes are markers for this cluster. So the cluster number 14 starts here, and we see the first most expressed gene is this one. So now we do one thing. We go to the already known markers that I told you before, the data set with all the markers from any cell type that I have on my GitHub. So let me show you, maybe, yeah, I think it's here. So you read the file, okay? The file is called like this. It contains many markers from both human and mouse. So if you open the file, you can see the first column is species. It tells you from which species the genes come from. So MM means mouse, HS means human, okay? So then you have the cell type column, which tells you to which cell type every gene belongs to. So for example, in the first row, we see that CTRB1 gene is a cell type for acinar cells in mouse and human, okay? So this data frame is really useful for any of you. It contains, again, many different cell types, so you can take a look at it. So what we do now is, because we don't know the we don't know to which cell type the cluster number 14 belongs to, we take the most expressed gene in this cluster, okay, and we look for the cell type that it belongs to, okay, in this data frame. Sorry, I have to read the file. Where is it? Ah, yeah. It's not here. Yeah, okay. So we see now that this gene, it is a marker for erythroid-like and erythroid precursor cells, okay? So it's telling you this gene belongs to this cell type, but we have to look for more markers, not just one, because one might not be informative. So now we look at the second one, okay, in cluster number 14, and we look to which cell type it belongs to, and it says again, it belongs to erythrocyte-like and erythrocyte precursor cells. So maybe we can look to another one, maybe this one. Again, we have to confirm with different markers, not just with one. Again, it says it belongs to erythrocyte-like and erythrocyte precursor cells. So now we know this cluster number 14 is this type of cell type, that the short name is like this, CESCs. So we rename our clusters like this, okay? Based on the markers we have seen, and we plot again all the clusters, and you can see now they have a name. We have annotated every cluster. We have given them the name based on these markers. And now we can extract different conclusions. For example, if we split the data again by sample, now we see that cluster 0 was macrophages. So we see that there are a lot of macrophages in the normal tissue and a lot of acinar cells and ductal cells, but in the tissue around the tumor and in the tumor, the number of these cell types is lower. But then, for example, we see that mast cells and fibroblasts and which more, B cells are much more expressed in tumor, which is normal because these are immune cells, and in cancer, the immune system is activated. So that's why we see a higher expression of immune cells around the tumor and in the tumor, okay, compared to the healthy tissue. So these are interesting things we are already seeing in the data. And so we can look at the different proportions, we can keep comparing between condition, etc. And then we can also analyze which genes are differentially expressed between conditions. So which genes maybe are upregulated between healthy pancreas and tumor in every cell type, okay? So we do that with these two lines here, okay? So we split the objects, and we then find markers between, in this case, I chose healthy and the tissue around the tumor, and we group by sample, and we choose the MAST method because this MAST method is really accurate to find differentially expressed genes for single cell data, okay? We also set the minimum log-fold change to 0.2 and only pos equal to true, okay? This is the way we find differentially expressed genes between conditions. So we run this and we get again a matrix, and we can see, maybe, yeah, it's okay. We can see, for example, if we want to focus on fibroblast, we do it this way. From the list we have created, we choose fibroblast, and we look at the differentially expressed genes of fibroblast between healthy and the tissue around the tumor. And we see the first one is this gene here with a log-fold change of 2.9 and a significant p-value. And if you guys look for this gene on the internet, you will see this gene is expressed in cancer-associated fibroblast, okay? Yeah, sorry? So with this, we are seeing that there are cancer-associated fibroblasts in the tissue around the tumor. And the same you can do for every cell type. If you select here that you want to analyze maybe the macrophages of whatever cell type, you can see which genes are upregulated between conditions, etc. And then you can save all this big file and taking a look at it more carefully. So actually, with single-cell RNA-seq analysis, you can do much more, okay? Because today we have a limited time, I just did until here, but you can do much more things that are also a bit more advanced, okay? But I think this is a nice introduction for you to single-cell RNA-seq analysis. These are some of the things you can do further, but this is, as I said, more advanced. I think for you guys, like for beginners that want to start with single-cell RNA-seq analysis, this is a good introduction, okay? And we can see many interesting things here already. So if someone has questions, I think now is the moment.